Great. So thank you very much to the organisers for giving me the opportunity to speak here. Especially thanks to BrightFocus. So I've been really, really lucky to have 3 grants from BrightFocus. I had one, the first original one was on AMD, the second one was on Alzheimer's disease, and then I recently had the honour of supervising, being the mentor for a postdoc as well. So I just need one of those glaucoma grants to complete the full Trinity, and I'm based in Trinity College in Dublin as well. It's, there you go. The glaucoma audience and reviewers are a bit trickier though, so I need to figure out how to manage them a bit better. So look, what I'm going to tell you today a bit about is the role of the inner blood-retina barrier in AMD. And some of the comments might sound a bit provocative because AMD is an outer retinal disease. So bear with me as I go through this. These are my disclosures. Trinity College does own some intellectual property related to regulating Claudin-5, this gene we're going to hear a lot about today. So they own some IP related to regulating Claudin-5 at the blood-brain barrier, but really nothing else associated with this talk has any link with any of these disclosures. So a bit of background on the current standard. So you go into a library in a university and take out a medical textbook and start exploring the vasculature of the brain. And you'll start getting images like this, you know, these types of medical textbook images of the large vessels supplying blood to the brain. And really, the human brain needs a massive blood supply because unlike other organs, the human brain can't store glucose, and that's the same as the neural retina. There's no capacity to store glucose in this tissue, so it needs a constant supply of glucose from the diet, okay? So in humans, we use about 20 to 25% of our daily energy intake simply to make your brain function. And because of that really massive energy demand, and because we can't store glucose, essentially every neuron is basically about 20 microns away from a capillary. So if you think about that and you make a section of human brain and look at it under a microscope, every time you see a neuron, you see a little capillary beside it. That capillary is perfusing and feeding that neuron with oxygenated blood, but also glucose. So essentially it implies that every neuron in the brain has its own capillary. And we can probably say that with the retina as well. The proximity of microvessels in the human retina to neurons is probably about the same. So really, if we take out the capillary network of a human brain and stretch it out side by side, it would stretch from Dublin to Paris. If we were to look at the surface area of those blood vessels, it would have the surface area of a tennis court. So 20 square meters is the surface area, okay? So when I look at a human brain or a human retina, all I see is blood vessels. And if you look at, this is a vascular cast of a human brain, okay? So what's been done here is a polymer has been perfused into a cadaver. It's been pumped around for about 10 to 15 minutes, allowed to set, and the brain is taken out of the calvarium, and then the brain tissue, the neural tissue, is digested away in an alkaline bath. And you're left with these incredibly dense network of vessels, okay? So when I look at a human brain, I see blood vessels. When I look at a human retina, I also see blood vessels, okay? So the blood vessels that we see in the human retina have properties that are absolutely identical to blood vessels in the brain. So it was mentioned earlier on that the retina is not an extension of the brain is an extension of the retina. They're neither. The retina is the brain. The retina is simply light-sensitive brain. That's the way I look at it, okay? So when we go to the literature and try to understand the integrity of these blood vessels in the brain in health and disease, we can probably apply a lot of the same biology to the retina because the retina is just light-sensitive brain. And the properties of these vessels in the retina are identical to the properties that we see in the brain. So let's talk a bit about those properties. So when people talk about the blood-brain barrier, sometimes there's a little bit of confusion. So, I know the vast majority of people here will understand what the barrier is, but let me just tell you first of all what the barrier is not, because there's sometimes a little bit of confusion. The barrier is not a seal around the brain. It's not a point at the base of the brain where things can get into or out of. The blood-brain barrier and this inner blood-retina barrier that we're going to talk a bit about is simply the description of the properties that endothelial cells that line these blood vessels have, okay? The properties that these endothelial cells have is that they've got very, very low rates of what we term fluid phase transcytosis. They highly regulate what gets into the brain or retina and out of it, okay? So essentially things that need to get into the brain or retina, they have receptors or transporters that allow them to get in. So organic ion transporters, glucose transporters. There's also junctions between endothelial cells that can tightly regulate what gets between the cells via the paracellular pathway. And very originally they're called tight junctions. And my lab does a lot of work in these tight junctions, and you're going to hear a lot about them. One final note on the endothelial cells. These endothelial cells that line the blood-brain barrier and the blood-retina barrier, if you take them out of the system and try to grow them in culture, they lose all their properties. So their barrier properties fall apart, the receptors that they express express start to decrease. And really, these endothelial cells are highly nourished by surrounding pericytes and the foot processes of astrocytes. Okay, so these pericytes churn out all these juicy neurotrophic factors that really nourish the endothelial cells. And it's really, really critical that we have these properties. So essentially, that word there that you see, brain, remember, retina is brain, so that's interchangeable. This is the blood-brain barrier and the inner blood-retina barrier. So my lab is very, very heavily focused on the tight junctions of the barrier, okay? And just cut a very, very long story short, these junctions are just proteinaceous junctions that are found between two contact points of two adjacent endothelial cells, or sometimes in these tiny capillaries that we see in the retina and the brain. A single endothelial cell can loop around a lumen and interact with itself and form a tight junction with itself. But by far and away, the most enriched tight junction component at the blood-brain barrier and the inner blood-retina barrier is this protein called Claudin-5. So my lab is, I don't know whether it's a healthy or an unhealthy obsession with Claudin-5, but this is our most favorite gene and protein. It's a small protein, 23 kilodaltons would be considered a small protein. It's a 4-pass transmembrane protein, which essentially means that it passes the plasma membrane 4 times. It has these 2 extracellular loops. Now these extracellular loops, they come out of the cell and they interact with 2 extracellular loops of Claudin-5 on an adjacent cell, okay? So they form what are termed homotypic interactions. There's so much Claudin-5 in a single human brain or retinal endothelial cell that we estimate there's about 18 million Claudin-5 molecules per cell. That's massive. If you think about the league table of enriched proteins in brain or retinal endothelial cells, you're talking about a protein that is expressed as high as GLUT1, as high as beta-actin. All the students, when you're normalizing your blots and your qPCR, you're using beta-actin. There's more Claudin-5 in a retinal or brain endothelial cell than there is beta-actin, okay? So these junctions are really, really tight. In 2003, Claudin-5 knockout mice were reported, and this is what they look like. So similar to VEGF, when you knock out Claudin-5, the mice are embryonic lethal. They get past embryonic day 10, so the animals do get born, but they die within 10 hours of birth. So it's a highly dosage-sensitive gene. The top image you're looking at here is a mouse that we've transcardially perfused with a very, very small molecule of about 400 Daltons molecular weight. You make a section through the mouse, and in a classic blood-brain barrier experiment, you see where the dye goes. So the dye is in a reddish brown. It goes everywhere in the little mouse's body, except for the brain or the spinal cord, because of the blood-brain barrier, because the properties of the endothelial cells are excluding that molecule from diffusing from blood into brain. Look what happens when you knock out Claudin-5. The molecule now goes everywhere. It floods into the brain, it floods into the spinal cord, it floods into the retina. Now, this paper was published in 2003 by a group in Japan, and they were really clever in what they did. They started to titrate upwards the size of molecules that they were perfusing. So they went from 400 Daltons to 800 Daltons to 1200 Daltons, and they discovered that the higher they went, the barrier was still intact. So Claudin-5 seemed to be able to Claudin-5 was able to exclude larger molecules, but not small molecules. So they come up with this idea that maybe Claudin-5 was really critical in regulating size selectivity at the barrier, the blood-brain barrier and the blood-retina barrier. If you go to the literature and look at blood-brain barrier disruption in diseases of aging, in loads of neurological diseases and psychiatric diseases, since the late 1950s, there's been an explosion of papers. So last year alone, there was over 1,000 papers linking blood-brain barrier disruption. Remember, these blood vessels in the brain that are identical to blood vessels in the retina because the retina is part of the brain. This morning, I checked how many papers were in PubMed relating inner blood-retina barrier disruption to aging of the retina, or AMD, and there were 10 papers, and 5 of them were mine. So there's a bit of a gap in the literature here, okay? So we really need to start exploring this concept of what is the role of the inner, I'm happy to admit if I'm wrong, that maybe the inner blood-retina barrier is not involved in any way in the aging retina and AMD, but this is what we're dealing with. We're dealing with 2 what are apparent disparate areas of the literature, but in my mind, they're actually identical. They're completely similar, and we should treat them as similar. So the blood-retina barrier. So people will know from the literature there are 2 barriers in the retina. There's the inner blood-retina barrier and the outer blood-retina barrier. But it's the same in the brain. We've got a choroid plexus barrier that makes CSF. The choroid plexus makes CSF. And those epithelial cells in the choroid plexus, they're identical to the RPE. So the brain is just like a big retina with the RPE attached, but the RPE is the choroid plexus epithelial cells. For the purposes of this talk, what I'm going to focus on today is just the inner blood-retina barrier, okay? Just these blood vessels that are similar to the brain microvasculature. The outer blood retinal barrier is made of the retinal pigment epithelium. They contain tight junctions as well. The most enriched tight junction component in the RPE cell in humans is claudin 19, not claudin 5. There's no claudin 5 in the RPE, but both of these barriers contain tight junctions, okay? So we could do another lecture, a whole series of lectures on the outer blood retinal barrier, but for the purposes of today, we'll just talk about the inner blood retinal barrier. So, this is what the retinal vasculature looks like. So it's really important to recognize that the blood vessels aren't everywhere in the retina. Okay? When you make a section of human or mouse retina, what you notice is that the vasculature, we can look at it quite simply and break it up into 3 distinct layers. So we have those superficial blood vessels. When you look at a fundus photo of a human or a mouse retina, you can see all the surface blood vessels, generally the larger caliber blood vessels. We also have this intermediate vascular plexus that kind of goes as far as the inner plexiform layer, and then we have what we call a deep vascular plexus. So 3 main layers of blood vessels, ones that sit on the surface and ones that dive deep into the depths of the retina. But these vessels only go as far as the outer plexiform layer. Okay? From the outer plexiform layer back to the apical surface of the retinal pigment epithelium, there's no blood vessels. It's completely avascular. Okay? But it's still got the same principle. The proximity of these deep vascular plexus capillaries is very, very close to the outer nuclear layer and the photoreceptors and rods and cones. And so we have this mantra in neurological disorders when we're talking about neurodegenerative disease. Because of the proximity of capillaries to degenerating neurons, when a neuron dies, eventually the small capillary that's feeding it will suffer as well. It'll start to show pathology. Sometimes it can happen the other way around, where there's pathology in the microvasculature that will drive the neuronal dysfunction. And to my mind, in retinopathies in general, we should be looking at this as well. What is the inner retinal vascular component to all of these degenerative retinal diseases because of the proximity of capillaries and blood vessels to neurons? We also have amazing technology in ophthalmology that we simply don't have in neurology. So we've got good high-resolution imaging from MRI, but really we don't have this detail of resolution that we can get in ophthalmology. This is a beautiful OCT scan showing the density of the vascular network in the human retina with the avascular fovea. So at the very, very center of the macula is the fovea, cone-rich fovea. There are no blood vessels, for good reason, because the majority of your visual acuity is being mediated by this high density of cones that can't be impeded by overlying vasculature. Now, a number of years ago, working with Christian Grimm here sorry, I was here, I'm not in Switzerland. I was in Switzerland on Wednesday. I'm now in Czechia. We were able to screen human donor eyes. So Christian had this bank of 28 human donor eyes that he had actually dissected out the periphery of the retina and the center of the retina where the macula is. And we asked a very simple question. We said, well, this gene claudin 5 is so critically important for maintaining size selectivity and permeability of the human retina. Is there a difference in the enrichment of this protein and gene in different areas of the retina? So what Christian was able to do, working with a very talented postdoc at the time, Federica Storti, was he was able to do qPCR of the central area of the human retina and the periphery. And straight away you could see that there was a huge enrichment of this component claudin 5. The transcript levels of claudin 5 were much higher in the center of the retina compared to the periphery. And when we do this technique that we're going to talk a lot about in a while is fundus fluorescein angiograms. When we take an individual, dilate the pupils, and then inject systemically sodium fluorescein, we light up the blood vessels. The nature of this molecule, sodium fluorescein, is that it's about 300 Daltons molecular weight. It's got very, very low rates of fluid phase passage across the blood-retina barrier, so it doesn't really readily move from blood into retina. And if you see a signal with fluorescein after you've injected it, it generally means the barrier is disrupted. So this is a normal, healthy human that's been injected with sodium fluorescein. And again, you can see very, very clearly at the depth of the fovea, you can see all these fluorescein-filled blood vessels. So this region in particular is absolutely chock full of claudin 5. There's loads and loads of claudin 5 in that region. So we go from humans, reverse translate back to mice. Where is claudin 5 expressed in a mouse? So we've generated these mice that allow us to produce green fluorescent protein from the claudin 5 promoter. So wherever claudin 5 is produced, we'll see fluorescence. Okay, so you can clearly see when you make a flat mount of an adult retina, you can see claudin 5 is enriched in the endothelium of the blood vessels. When we peel off the neural retina and stain the eye cups, you see that there's absolutely no fluorescence. There's no claudin 5 in the RPE. This is stained for ZO1. Okay, so there's only claudin 5 in the blood vessels. When we make sections This is like the schematic, the diagram I showed you. Look, you can see the blood vessels here, the superficial, the intermediate, the deeper vascular plexus. This is Claudin 5 again, the Claudin 5 GFP mice. There's no blood vessels from here back to the RP. And to Rob's point, there is actually Claudin 5 in the choroid, in a mouse. So that is interesting that there may be this interplay as well of junctional material in the choroid that may contribute, as we'll see, to AMD. But really, that is the pattern of Claudin 5 expression in the mouse retina. Now, I don't need to talk to this audience in detail. We've had some brilliant overviews of AMD from Usha and Tiernan this morning. But really, you know, maybe using this terminology of it's an epidemic and epidemic proportions is a bit too strong. I sometimes convince myself that it was a bit too strong. But really looking at the numbers now of patients who have AMD in Europe, really it is getting to this stage of it is almost at epidemic proportions. There's huge numbers of people living with this condition. And obviously as we keep getting older, the numbers are simply going to increase. So this is what early dry AMD looks like in Ireland. Doesn't look much different from other parts of the world. The pathognomonic signature, obviously, of AMD is the number and presence of drusen in the macular area. This is what GA looks like. So this is one of the individuals that was involved in our clinical research with this large spot of atrophy. And really that individual would have had a huge amount of difficulty reading books and seeing faces when they were visiting the clinic. But if we take out the eye post-mortem and look at what these retinas look like inside profile, this is a normal retina, so a non-diseased retina. Okay? The structure is pretty normal of the outer retina and the inner retina. But in areas of GA, you always see these, at the margins of atrophy, you see these fat vesiculated RPE cells. And in the areas of atrophy, you see no RPE cells because the RPE is atrophied. Now, really, in this context, we've always wanted to know what's happening in here. What's happening in the inner retina at the margins of atrophy and at late stages when we actually have retina that's overlying atrophic RPE. What is the integrity of the blood vessels that is in the inner retina? And is that in some way even contributing to the disease that we haven't seen yet? In 2019, this paper came out. It was a small paper in experimental eye research, but it really caught my attention. It was from Josh Denave's lab. And they showed in a very small number of donor human AMD eyes where they peeled off the neural retina and just looked in the neural retina. They did these Western blots for albumin, for fibrinogen, and for IgG. And what they saw was a significant increase in albumin levels in the retina. So these were atrophic. There weren't neovascular AMD. Atrophic, dry AMD retinas had high levels of albumin. Okay? Now, albumin should be inside blood vessels. Okay? You should really not see a huge amount of it in the extravascular space. But what caught my attention was there was a significant increase in albumin, but not fibrinogen or IgG. Now, albumin is small in the context of these systemic molecules. Albumin is a molecular weight of about 70 kilodaltons. Fibrinogen is about 360 kilodaltons. It's huge. So if you see any extravascular fibrinogen, it means you've got a catastrophic breakdown in the barrier. And IgG, similarly, is quite large. It's about 120 kilodaltons. So as soon as I saw that, I said, there's size selectivity going on there. The barrier is disrupted enough to allow albumin to get out, but not the other components. So back to the Campbell lab's favorite protein, Claudin 5, what would Claudin 5 be doing in this type of scenario where molecules that are small enough are getting access to the neural tissue, but to the exclusion of larger molecules like fibrinogen and IgG? So what I'm going to shift to is to talk to you about some data largely from my lab. As I said, there's only 10 papers really on the inner retina and AMD. So a lot of this work started probably about 7 or 8 years ago. And it started with this discovery that we made that we came across this site that had a circadian rhythm database of all the genes that cycle in circadian rhythms. And just for fun, we said, well, what does Claudin 5 do? This really critical component of the blood-brain barrier and blood-retina barrier. And we discovered that Claudin 5 cycles in a circadian rhythm in every tissue we look at. So if I gave you any gene and said, go to this database and look to see, does it cycle? About 30 or 40% of the genome does cycle in a circadian rhythm, but very few, if any, genes cycle the same way in all tissues. So every tissues that we looked at, from the brain tissues subdivided into the neuroanatomical areas to systemic tissues, they all cycle in the same way in that in the evening time, transcript levels of Claudin 5 are really low, and in the morning, they're really, really high. So what we did was we decided to go back to our animal models, and we did a Western blot. And sure enough, when you do a Western blot for Claudin 5 in the morning compared to the evening time, there's low levels in the morning and there's high levels in the night. Okay? There's these really neat techniques you can do when you're doing circadian rhythm biology. You can dark adapt animals, so you can keep animals in the dark for 48 hours. If something is truly circadian, the shifts should stay the same, and we saw that. So in the evening time, Claudin 5 levels were low, in the morning they were high. And we can also invert the rhythm, so we can change the light cycle of the animals. Similar to people here who've come from the US and might be jet lagged, your circadian rhythms are gradually changing. So it takes about 3 weeks to completely change the circadian rhythm, but sure enough, animals that were inverted, Claudin 5 was inverted as well. So Claudin 5 was a circadian gene, and it was a really profound discovery that we discovered that this really, enriched component of the blood-brain barrier and blood-retina barrier was actually a circadian component. Now, what we decided to do was we decided to do fluorescein angiography to see whether or not this phenotype of Claudin 5 cycling was matched with an actual physiological readout. So if we did a fluorescein, would we see any differential in the fluoresceins in the morning compared to the evening? And believe it or not, at the time, there was no good quantitative way of objectively quantifying fluorescein scans. So we developed one ourselves. It's called FOVAS, Fluorescent Ocular Vasculature Analysis Software. So if you want to know more about this, Jeff O'Callaghan, who's a postdoc in my lab, is here, so he can talk to you in detail about this. But really what it was we wanted something that would be pan-species, so that basically we could use the same software in a mouse, in a non-human primate, but also in a human, and we'd have confidence that what we're seeing actually transcended the species. So this is what it looks like. This is a fluorescent angiogram in a mouse, an adult mouse in the morning time, which is unusual. I mean, it doesn't look unusual. It's a normal fluorescent angiogram. And the evening, it doesn't look unusual either. But when you quantify these fluorescent angiograms, you see that the leakiness or the signal is higher in the evening time compared to the morning, okay? So it's not a pathological fluorescein leakage. It just means that the signal that's being produced by the fluorescein is higher in the evening and lower in the morning. And to our minds, what's happening here is that fluorescein signal that's higher in the evening is fluorescein that's in the interstitium of the retina. It's moved from blood and gone into the retina. So what is the purpose of this Claudin 5 cycling? So what we're thinking of, based on our knowledge of the brain, is that maybe it's involved in replenishment, so replenishing spent metabolites. So maybe it's involved in photoreceptor replenishment, outer segment replenishment. The passive diffusion of metabolites, lipids, fats that are coming in from the diet, allowing them to again, replenish the retina. And we, talk about this thing, retinal interstitial kinesis. Is there something that's similar in the retina compared to the brain? We know in the brain field we have got this glymphatic system hypothesis where fluid moves through the interstitium of the brain. That is very, very poorly defined in the retina, so maybe Claudin 5 is intimately involved in that, allowing fluid to move via the interstitium of the brain. So what I'm going to talk to you about is 4 different models that we've used to try to figure out, what actually is the role of Claudin 5 in the retina, and is it in some way, linked to fluid dynamics and movements, but also maybe to phenotypes that we're familiar with in AMD. So the first model that I'm going to use, and again, no model is perfect, that's why we use multiple models, speaking to Rob Mullen's point. The first model was an adeno-associated virus that we made. So this isn't a therapeutic virus, this is a pathological virus we're making. So we're inside the genome of the virus, we've put a hairpin RNA targeting Claudin 5. So it means that when we inject the virus, the animal basically will have low levels of Claudin 5 as the shRNA is produced, okay? So in the animal's right eye, we injected an AV targeting Claudin 5, and the left eye, we injected a non-targeting virus. We showed again with Western blots that we can decrease Claudin 5. So this eye here has very, very low levels of Claudin 5. This is an MRI scan, by the way. And we sometimes use MRI instead of fluorescein because it allows us to look at the left eye and the right eye absolutely simultaneously. Instead of fluorescein, we use gadolinium. It's a slightly higher molecular weight dye. And this is what it looks like. So when we suppress Claudin 5, we get this beautiful phenotype. The right eye is leaky, the retina is leaky there, and the non-targeting eye is quite normal. And this is what the fluoresceins look like. So what we do is we inject mice in their left eye with a non-targeting virus, right eye with a Claudin 5 targeting virus, and we put them on a normal diet. So you can see an enhanced fluorescein signal compared to the contralateral eye. But that's a normal diet. But look what happens when we expose the animal to a really lipidaceous, high cholesterol diet. It looks very, very different, okay? So there's lots of focal points of fluorescein damage. Remember, we haven't touched the choroid, we haven't touched the RPE, we're simply targeting Claudin 5 in the inner retina. And this is what the retinas look like. So they look very, very similar to the margins of geographic atrophy that we see in human subjects. We get these fat vesiculated RPE cells. We see these autofluorescent RPE cells. So it kind of looks quite similar. So we're driving RPE cell death by targeting the inner retina. Okay? So this hadn't been shown before. We're not touching the RPE. We're doing nothing to cause damage to the RPE. We're simply targeting vessels in the inner retina and driving a pathology that looks like AMD. When we do flat mounts in the tissue, this is what it looks like. So we see these lovely areas of ZO1 positive RPE cells, but we see these islands of atrophy. And I don't tend to see this in models in the literature of dry AMD. Normally the models of dry AMD are very patchy, but what we see is focal points of RPE death, and we have a margin of atrophy. So what I think is happening here is that by inducing a focal point of inner blood-retinal barrier leakiness, we're then propagating the enhancement of material from the inner retina back to the outer retina. So we're flooding the RPE with toxic material. That's the hypothesis that we think we're moving along. Again, no model is perfect. So the second model that we've used is a mouse model that is heterozygous for Claudin-5. So it only has one Claudin-5 allele. Okay? So you can see in these mice when we do fluorescein angiogram, they've got a leaky blood-retinal barrier anyway, because remember, they're only producing half the amount of Claudin-5 as wild-type animals. So when you do fluorescein angiograms in these mice, you see this lovely signal. But similarly, if we take these mice, these Claudin-5 heterozygous mice, and feed them this high cholesterol diet, again with the idea that we're forcing lipidaceous material into their retina back to the RPE. We're overloading the RPE with material. This is what we see. So this is a long-term, a chronic challenge with a high cholesterol diet. And what we actually see here is focal points of damage, again, very, very similar to geographic atrophy, but we see a collapse in the external limiting membrane. This is a really classic feature that we see in geographic atrophy in humans, and this is what we see. Claudin-5 het mice on high cholesterol diet for 20 weeks, the external limiting membrane collapses, and they get focal points of RPE cell death. This is what it looks like in some of the clinical research that we do. A patient who has geographic atrophy, you can see a phenotype absolutely identical to this, where the ELM is collapsing, and you're getting the underlying RPE is atrophic. Model number 3, mice don't have a macula. So we all know that, and so obviously you can't just do non-human primate work at whim. Unfortunately, we have to go all the way to St. Kitts and Nevis in the Caribbean to do these studies. We work with this incredible species, the African green monkey. They're a non-indigenous species to St. Kitts and Nevis. And we work with a company called Versio, Matt Lawrence. We've got an academic collaboration with this group. And it's been really, really fruitful collaboration where we're kind of tagging on to a lot of the work that they're doing and to address the 3 R's of refinement and reuse, we're kind of using animals that are being used for other studies as well. So doing things like fluorescein angiograms that aren't too big an ask. This is the AM-PM differential that we see in the non-human primate retina. Okay, so any clinicians, if you look at these, they look very, very similar to human FFAs. They're almost identical. It would be difficult to tell the difference. But you can actually manifest these differences. So these animals will be anesthetized during this. You can see a differential even just by eyeballing it. You can see that in the evening time, it's brighter compared to the morning. We always leave time for the fluorescein to wash out, so there's no concept here that there's a carryover from an earlier fluorescein imaging. And this is the quantification. So when we use this FOVAS technique to quantify it, we can see that in all the areas of the macula that we look at, in the evening time, there's brighter signal in the evening compared to the morning. Now, what we can do in these animals is we can do the same experiment that we did in the mice where we inject the Claudin-5 AAV or the non-targeting AAV, but amazingly, because the macula is so large in these animals, obviously in humans as well compared to mice, we can inject an AAV into the superior macula and the non-targeting AAV into the inferior macula. And what you can see here, again on fluorescein, with the Claudin-5 AAV injected up here in the superior macula, you get a huge focal point of fluorescein leakage. Again, we're making the inner blood-retinal barrier leaky to see what will it do to the RPE. And this is the non-targeting. You can see very, very little signal from the non-targeting AAV. This is an OCT directly over the non-targeting region, which looks structurally completely normal. And this is an OCT directly over the area that has the inner blood-retinal barrier disrupted using the Claudin-5 shRNA producing AAV. So again, it looks very, very similar to what we see in AMD, where you get these vesiculated and stressed RPE cells. And again, this is the type of histology that we see with these fat, vesiculated, sometimes transmigratory RPE cells. Again, only by targeting the inner retina, not going near the outer retina. So the final model is human. So what we were able to do a number of years ago was get ethical approval to do fluorescein angiograms, circadian fluorescein angiograms in the morning and again in the evening in the normal non-diseased human. And so this is data that's representative of 35 normal non-diseased individuals over the age of 30 years old. Again, AM, we asked the people to come in as early as they could. We did a questionnaire that basically was able to assess what their deepest phase of sleep would be to allow us to normalize the data. And we then said, well, based on your deepest phase of sleep, you've got to come in for your evening scan at 8 or 9 or 10 o'clock. So we had access to the clinical research facilities to do this. And again, there's not much differences when you eyeball this, but if you concentrate on some areas, around here, for instance, it's brighter in the evening time compared to the morning. And when we quantify this, again, in every area of the macula, the signal is higher in the evening compared to the morning. Now, what does that look like in AMD and age-matched controls? We did the same study in healthy age-matched controls, and this is what we saw. So immediately looking at this, if I was to very quickly go back, look at those images and go forward, immediately in the aged images, when you put them side by side, the fluoresceins look brighter. So with age, in all likelihood, the vasculature is getting a little bit weaker, and so the signal is higher. That's what we believe is happening here, that the vascular tone diminishes with age. And so what we see is the cycling stops. So with age, you don't get the circadian cycling in the phenotype anymore. The signal is neither brighter nor weaker in the evening compared to the morning. We also see this in AMD patients. So we recruited patients with early and intermediate AMD, and again, that circadian signal is being lost. So we're trying to contemplate this idea that the inner blood retina barrier is becoming subclinically leaky in patients with early to intermediate AMD, and that in tandem with their background of smoking, genetic etiology, diet, could actually be the key driver of pathology that we see that's classically associated with AMD. So the take-home message here, what we think is happening here is that in the normal healthy retina and brain, Claudin-5 cycles routinely in these 12-hour cycles. It goes up and down and up and down. That's probably aligned with replenishment and allowing material to move from retina or brain back into the blood. With age, that likely slows down in some individuals. Maybe there's accelerated processes where this whole concept of Claudin-5 cycling ceases really, really early on, again, a background of genetic etiology or poor diet or smoking history that then allows for the inner retinal toxins to drive the RPE damage. So really some of the unanswered future questions that we have is, what is the actual contribution of this barrier disruption to the rate of GA development? So we haven't even done that yet. We know that there's a bit of a hint, a glimmer of its association with the onset of early AMD, but maybe what is its contribution to GA? Can quantitative FFA be used as a biomarker for dry AMD progression? So can we take individuals who don't have vision loss yet, but have a very, very high signal on fluorescein angiography when we quantify it? Are they at higher risk of more rapid progression to end stages of AMD? Does this inner blood retina barrier disruption preferentially drive the pseudodrusen formation? So we've got a hypothesis that a lot of the pseudodrusen that we see is actually coming from the inner retina. It's not coming from the choroid, it's coming from the inner retina. And could this inner retinal vascular contribution be a driver of subretinal drusen accumulation? And then finally, can this barrier regulation be used to stabilize the rate of geographic atrophy? So can identifying therapeutic targets like Claudin-5 and regulating its integrity prevent or slow geographic atrophy? So all burning questions. These are the references and further reading. As I said, a lot of them are from my own lab, but there are some other groups working in this area as well, especially Josh Geneve's lab. And finally, this type of work takes an absolute village to run, none of which I did myself. I'm just the representative of the lab as the head of the lab. So really, really thankful to all the funders and all the PhD students and postdocs. Some of them are here, especially Natalie, Jeff, and Avril, who have contributed to some of the work that I showed. Christian Grimm's lab as well, Federica, who's now in Roche, I believe, and all the clinical team that we work with in the Eye and Ear Hospital. And hopefully we have plenty of time for questions.
Thank you, Matt. Here's a question over there. If you have a Claudin 19 knockout, would it be the same phenotype?
Don't know. As I said, that's for a different lecture, Florian. Outer blood retina barrier.
And this is Yuan from University of Pittsburgh. It's a really nice talk. I also have the same question, but I also want to ask: have you tried overexpression of Claudin-5? Do you see any protective effect?
That's a great question, yeah. So that really is what we're focusing on now for the therapeutic regulation. We have used AAVs to overexpress it, but the AAV technology is just too good. It produces too much. So there's a happy medium with Claudin 5. It's like anything really. Too much of it is as bad as too little of it. So I think the AAVs are too potent at overexpressing Claudin 5. There are some drugs, a paper came out a number of years ago, actually from our collaborators in Roche, showing that a drug called Repsox, it's a TGF beta receptor antagonist, and when you use that, Claudin 5 levels specifically increase. So I think a gene therapy-based approach is probably too potent for overexpressing Claudin 5. You need to find that Goldilocks zone of a small molecule or some other way of regulating it, or just dialing down the AAV, like injecting very, very low doses. But it's a great question.
Thank you.
Hi, Ruchi from NAI. Great talk. I have a question. So you showed Claudin 5 knockout in a retina caused RP degeneration.
Yep.
Do you think it's driven by complement pathway?
Yes. Yeah. So we have RNA-seq data that shows that complement will manifest. So complement will manifest in the RPE and the retina after you suppress Claudin 5 and put the animals on a high cholesterol diet. So to our mind, that this vascular contribution, the inner retinal vascular contribution, maybe the reason we haven't really seen it or focused on it before is because it's a really early stage. It's almost like the Kickstarter for the damage that we see. So yes, we do see complement, and we can share that data with you as well.
Yeah, no, I think this will be, since, until now it's also been thought that complement is always on the choroidal side. So it will be nice to know that it could be acting on the apical side too.
Yeah, this is causing it as well. And it is kind of, the learnings that we have from the neurological community is that you focus on the pathology that you can see. So in Alzheimer's, we see plaques, we see it's a tauopathy, but really the clinical trials and the approved drugs that we have now for Alzheimer's, aducanumab, donanemab, they really aren't great, So whatever is happening much earlier, and we just can't see it in the end stage disease.
Hi, Lindsay Chu, MD PhD student at Duke. I was wondering if you could tell us more, is that since we know that there are many AMD patients who still have 20/20 vision, and so you were mentioning the possibility of using this FFA marker, and I'm wondering about the status of any kind of drusen in those early AMD patients that you're thinking of, and whether they have any visible findings on like dilated fundus exam, or if you're thinking this might be good for patients who are even before that stage.
Yeah, I think using fluorescein as a biomarker, I think, I'm not a clinician, so I take my guidance from my clinical colleagues. Really, I think it will be of use for very early or maybe intermediate AMD if we can have good longitudinal studies from now on. We do see, obviously there's patterns of how drusen appears with fluorescein sometimes. A lot of the reticular pseudodrusen, you do kind of see this halo-in effect sometimes around the focal point. When you see it on OCT, you see the fluorescein build up around the actual subretinal drusen body. So yeah, I think it's unknown yet. I don't have a clear answer for you yet, but longitudinal studies with fluorescein will address that. And this is the issue with fluorescein. The fluorescein has kind of fallen out of favor with a lot of groups because OCTA is very valuable to see the structure of the vessels. But really, to my mind, it's a functional molecule that you're injecting. You're injecting a molecule that its properties of how it interacts with the luminal surface of endothelial cells. And so the readout that you get from it's a very, very biological signal that you get from fluorescein that you don't necessarily get with OCTA. And I've got no affiliation with any company selling FFA.
Hi there, it's Rudy McGaw here from the University of Edinburgh. Thanks for a great talk. I was wondering if you know what is governing the diurnal variation of Claudin 5, and might it be the blue light ganglion cells?
Yeah, it's a great question.
And then the other, just a very quick question is, does the increased permeability allow toxins to go the other way at night time?
Yeah, so that's a great question. So we've done some work, we haven't published it yet, and the promoter region of Claudin 5 is replete with what are termed EBOX sequences. And EBOX sequences are a binding site for BMAL1, which is like a master regulator of transcription, master transcription factor, which is a regulator of circadian rhythms. So we have data to show that BMAL1 can interact with these EBOX sequences. So certainly that's how Claudin 5 is going up and down, because BMAL1 goes up and down based on the levels of melatonin and cortisol in the system. So that's pretty clear. The second question of is material moving out? That is the argument for the glymphatic system hypothesis, which is exceedingly controversial in the brain field of what is it, what's happening. That the glymphatic system hypothesis is basically this concept that the interstitium in the human brain increases in size during phases of deep sleep to allow for clearance of spent metabolites from the brain. So it's the biological rationale for why we need sleep. It clears the brain, washes the brain of toxins. And so the exact same principle is likely happening in the retina, that during phases of deep sleep, that the interstitium is likely getting bigger, claudin 5 levels are low, to allow for material to move across its own concentration gradient from retina back into the blood. So that doesn't all have to go out via the RPE and choroid and choriocapillaris. So it's a really, really good point. That's what I think is happening. Don't have proof of it yet, though.
Hi, Trevor McGill, Oregon National Primate Research Center. I've kind of a complex question, so it's a little bit biphasic, if you will. So
Okay.
The first part is, in your non-human primate studies that you did, can you tell me two things? One is, what was the route of administration, the surgical route of administration, and the time point that you looked at the expression of the claudin 5 GFP?
So the route of it was an AV9, so it was subretinal in both cases. So that the superior and inferior macula, it went subretinal, AV9. And the length of time, I believe, was 48 days or 42 days. I don't have the exact number, but it was more than a month.
Okay. So the one thing that I would say, it's interesting because sometimes when people will even do just a subretinal injection with saline, you can get up to many weeks of still fluorescein leakage just as a consequence of doing a subretinal saline injection. And so it would be interesting to know how that differs from each of your targeting and non-targeting AAVs.
Sure, we see the exact same thing with that as well. And that's why using the same eye for same titer, same serotype of AAV in superior and inferior, what we do is you switch up the inferior. So sometimes you put the non-targeting on top, the claudin 5 on the bottom, but we see the same effect as well. That is the beauty of using the one eye for both AAVs for the subretinal. But we do, that point that you're making, you do see a focal point of RP atrophy when you do a subretinal injection in monkeys.
Hi.
We can follow up after, yeah.
Danielle Little. Wonderful talk, very interesting. I was curious, so a similar question was in my mind about the route of administration. So if it is subretinal, do you think there's any chance that you're actually targeting the choroidal claudin 5? And if you are, have you done that deletion or compared the downregulation of claudin 5 in the inner versus the outer?
Great question. No, so we haven't done that. I would say that when you inject AV9 subretinally, you don't, to my knowledge anyway, you don't transduce the choroid. Certainly I haven't seen it with reporters. We did use, to address that complete issue, we did use the heterozygous mice on the high cholesterol diet to try to figure out, was there a differential there? So I do think that the atrophy is likely coming from the inner retina based on the fact that the subretinal AV likely does not transduce the choroid. Trying to do it the other way around of just exclusively transducing the choroid, unfortunately, it's a tricky tissue to get exclusive access to. Maybe in larger animals, with suprachoroidal delivery, but they're expensive experiments, and take your point though.
So bad news is we ran out of time, but the good news is he's really quite nice and you can talk to him with coffee.